Okay. All right. Oh, like an open today's lesson, right? That would be good. Not 10. We're 11. We're in chapter 11. Oh, but I want to be in the other one. Yes, ma'am.
For the lab final, why did internet time if it's online?
Yes.
Okay.
Yes. So send me an email reminder.
Okay.
And what I'll do is I'll just build it into the exam online. I'm gonna do exam online.
Yeah, because I know I can't take it in there because they're closed. I thought.
Well, right. But
Yeah.
do you want to take it there the next day? Like if it is better there, but it'll still be a Moodle.
Yeah.
It's up to you. Just think about it.
Yeah.
And we can talk about it in lab.
Okay.
Think about it. Yeah. Okay.
Lab final. I mean, not final. Reports not due this week, right? It's next week.
Correct.
Okay.
Correct. Correct.
Oh yeah.
I got 2 of them.
Okay. How we doing?
It's Wednesday.
It's Wednesday. It's Wednesday, and what is the next time I see you?
The test.
An exam on chapters 9 and 10 only. So what we're going to cover today is not on the exam. Okay. But it will be on the final exam, which is, very soon anyway.
I think you answered this already, but just the final gonna have a big chunk.
So yeah, so I answered this yesterday, but it's okay. It's good to reiterate. So because this content, chapter 11 content, is not going to be on exam 4, right? There will be a larger portion of the final exam that has chapter 11 material versus all the other chapters, right? So I'll do chapter 11 and then I'll fill the rest with chapters 1 through 10 pretty much equally. Be okay. It'll be okay. It'll be good.
Did you decide how you're going to do the percentage for lab?
I'll talk it in lab. We'll talk about it in lab. Absolutely. Anybody else for exam this Friday, right? We're all good on that. Okay. Good. All right. I feel like I had something else to tell y'all, but I don't remember what it was. Oh, a joke. See? A joke. Let's see. It doesn't. There was a really good one this morning. What was this morning's? Because I have one, but it wasn't really good. But I really liked this morning's. I can't remember what it was. Oh, man. All right. What did the dog say when he sat on sandpaper? I said that yesterday? Oh, okay. Let's see. Oh, no, I got so many more. It's okay. Oh, I remembered it. What did the turkey say to the farmer on Thanksgiving Day?
What?
Quack, quack. He doesn't want to get eaten. He doesn't want to get eaten, so he's going to pretend to be a duck. It's kind of cute, right? All right. So, I did not want to do that. All right. So before we talk about oxidative phosphorylation, I want to do a quick little review, which this will be good for you. This will be really good for you for the exam. I'm going to tell you that from one glucose molecule that I can get a maximum of 32 ATP. That's a little new piece of information, right? Because we've talked about glycolysis and citrate and how many ATPs, and I'm going to count GTPs as ATPs. So how many in citrate and glycolysis? How many do we get? Oh, y'all, you should know this. How many ATP in glycolysis? 2. Y'all with me? Okay. How many we get in citrate? 1 per turn. How many turns per 1 glucose molecule? 2 turns. So how many am I going to make? Right. So this is 2 turns. Okay. So this is really only 4 ATP. That's what? 12% of the total ATP? Did we generate lots of ATP yet? No, we really haven't. So where is that other 88% of the ATP going to come from? Electron transport chain, oxidative. No. Phosphorylation gives us the remainder. So what do we have left? 28. And it's about 28 because it depends on what cell type you're in, right? So it gives us about 88% of the ATP. So where are we getting the energy to undergo this oxidative phosphorylation? What other high energy molecules do we make in glycolysis and citrate other than ATP? NADH and FADH2. I would like to map where we make them and how many we make them. Think y'all can do that? Yes, you can. I know you can. Okay, glycolysis. Glycolysis. We're going to start out with one glucose, right? That one glucose in glycolysis gets broken down into what? 2-pyruvate. 2-pyruvate. What else? What are my other products? Y'all told me 2 ATP molecules, right? What else? Y'all should know this. Y'all should really know this. What else do I get? You get, we said 2 ATP, 2 something else, 2 NADHs, right? Okay. So if we're quantifying what's going to happen with them, right? This process takes place where? Cytosol, right? So do we have oxygen or not? No oxygen. Okay. So what's going to happen is we're going to change cellular compartments. That's going to be my little, we're moving, right? We're moving from the cytosol into where? Into the mitochondria. So we're going to take our 2-pyruvate and we're going to ship them into the mitochondrial matrix, right? Really beautiful drawing of the mitochondrial matrix. You do, but those aren't energy carrying molecules, but you do make them. Those are products, yes. Okay, so we go into the mitochondrial matrix, right? We take that pyruvate and change it to what? We change it into acetyl-CoA. Can I move that? What enzyme does that? Okay, so when we convert from pyruvate to acetyl-CoA, we don't just make acetyl-CoA. What else do we make? How many NADHs? Because really we make 2 acetyl-CoAs, right? How many pyruvate? So scroll up. How many pyruvate? 2. So we had 2 pyruvate going to 2 acetyl-CoA and 2 NADH molecules. Okay. So these NADH molecules that have been made, what are they going to do? Well, these are in the mitochondrial matrix, right? These 2. So these 2 are in the prime spot to go through the electron transport chain, right? Once they go into the electron transport chain, you're going to use them to generate ATP. And we'll talk about how many in just a second. But that's the point, right? We go through the electron transport chain and we convert those energy carrying molecules into ATP eventually. That's the whole point of oxidative phosphorylation, right? What else do we make? What else happens in general? Because we're kind of done with it. Once you get to ATP, you're kind of done, right? You're kind of done. So what do we do with acetyl-CoA? Acetyl-CoA goes into citrate. So what are we going to make in the citrate cycle? Remember, this is going to be how many rounds of the citrate cycle? 2, because we have 2 acetyl-CoAs. So for every 1 go round, how many NADH do I make? 3. So I got to double that and make 6. And those are then going to go on to the electron transport chain, and we're going to make ATP out of them, right? Okay. Now, what else do I make? In 2 rounds, I make 2 FADH2. Those are going to go through not the whole electron transport chain. Those only go through part of the electron transport chain. So what we're going to see in a second is that that's going to make a little bit less ATP, but it still makes ATP. Okay. Then what? We also make how many GTPs per cycle? 1. So that's a total of 2. So I guess we'll put a plus 2 GTPs, which convert to 2 ATPs, right? Okay. So the question is, when we take these energy carrying molecules, how do we turn them into ATP, right? Well, they go through the electron transport chain. There's a conversion for knowing how many ATPs are made from NADH and FADH2. So we know that we can make 5 ATP molecules per 2 NADH in the electron transport chain. We also know that we can make a few less, so 3 ATPs per 2 FADH2s. Okay, so if we have 2 FAD molecules here, how many ATP am I going to generate? If I have 2 FAD molecules here, how many ATP am I going to generate if I go through the electron transport chain? 3. Okay, if I have 6 NADHs here, how many ATP am I going to make? Ooh. We're going to make 15. So Courtney, I'm sorry, I didn't see your question until just now. Did I answer that?
Final products are 2 cycles.
Well, yeah, everything, but I don't know when she asked that. That's the question. Like at what point? So
Yeah, just now.
oh, just now. Okay, so this is really 2 turns of the citrate cycle, because what are we starting with? 1 molecule of glucose. So 1 molecule of glucose goes through glycolysis once to get 2 products of pyruvate. Those 2 pyruvate, you have to go through citrate twice in order to use up that 1 glucose, right? So everything's really based on 1 glucose. All the math, everybody's math in every book is always based off of 1 glucose. Okay, I'm going to try to, I wish this box were just a little bit smaller, but it's okay. All right, so 2 NADH here. What do we make? Another 5 ATP. Golly, that's a lot of ATP, right? Aren't we making a lot of ATP here? Now, how about these 2 NADHs? Well, not quite. That's the problem. The problem is these 2 NAD molecules are in the cytosol. And so actually what has to happen is they have to be shuttled into the mitochondrial matrix. And it depends on which tissue type, what kind of shuttle system you're going to use. So these 2 ATP will just make 2 ATP, right? But this NADH can go one of 2 ways, right? They both will eventually make ATP, but because there are different shuttles, we're going to make different amounts of ATP. So if you're in the liver, you're going to make the maximum amount of ATPs, you're going to make 5. But in muscle, you're going to make a little bit less and you're going to make 3. But the benefit to that is that the muscle shuttle system is way quicker, right? So what happens when you're exercising? Is it more important to make more each round or to be able to go through more rounds of shuttling? More rounds of shuttling, right? Because you have different intensities of exercise, but your liver is kind of stationary, right? So it doesn't have to be really quick. Okay, so what do we get for a total? It's going to be different if you're in the liver or the muscle, right? So don't count 5 and 3, count either 5 or 3 so that you have a total for the liver. Let me do this in a different color. Let's do the liver in blue and the muscle in red, right? So total them up. What do we get? No, I just said that. Muscles red? Yeah, muscles red. Liver. What do we get when we total these? Because it's going to be different. Total up your ATPs. You're going to get more in the liver, right? But you'll be able to regenerate more ATPs faster in the muscle. So liver makes how many? 30. Oh, I'm wrong. Should be 32. Muscle makes 30. Muscle makes less, right? Because if we look here, our muscle's only making 3. Our liver makes 2 more. Yeah. Yeah. Okay. So it's really, I want to start with this because this is the big picture of where we're going. This is why the electron transport chain is so important, right? We've done all this work in glycolysis. We've done all this work in citrate, but we've only made 4% of the ATP. So oxidative phosphorylation really provides us with the majority of ATP for the body. And the theory behind how this works was really revolutionary at the time. And so it just made it, I think, that much more interesting. Okay, so we understand how glycolysis linked to citrate linked to electron transport chain, right? Okay. All right, so let's actually, let me find, how do I get there? All right, so this is what we're actually going to do today. And I want to put my chat back up now that I've moved it. Okay. All right. So how does the electron transport work? Well, it's through Chemi osmosis, right? So it's the Chemi osmotic theory. It's the movement of protons. It's really cool. And so we'll look kind of at the theory in general and then exactly how the electron transport chain makes that happen. And then at the very end of the electron transport chain, what are we supposed to generate? ATP. So the next thing that we're talking about is the structure and the function of the ATP synthase complex. The last one, not the last one, but the fourth section is those 2 transport systems that we were talking about for NADH and FADH2, right? So those, and then 5 is regulation. So I don't know that we will get to 4 and 5. We will see. We will see how far we get. But it's more important to me that you understand up to 3 than that we get 4 and 5. Yeah, okay. All right. So the chemiosmotic theory was, like I said, really, really revolutionary. But the thing is that it's conserved across all different organisms. So you see it in eukaryotes, you see it in bacteria, right? So that's how we know something's very highly conserved. If we look at very complex things and we look at very simple organisms, do they have the genes that code for the proteins in the process, and are they conserved? And they are extremely highly conserved. And so we know that all kinds of different systems use this. And so this is really like one of those really big unifying principles in biochemistry, right? So this is a super, super important concept. So when we left off on the citrate cycle, where were we? Where were we making the products of the citrate cycle? In the mitochondria, but we know the mitochondria has multiple things, right? It has an outer membrane, it has an inner membrane space, it has, so where is it in the mitochondria? In the matrix. We're in the mitochondrial matrix. So citrate cycle is happening in the mitochondrial matrix. So this chemiosmosis is going to also have to happen in the mitochondria. And we're going to look at the different parts of where it's happening. So once the electron transport chain becomes active, it creates a proton gradient. And that proton gradient is across the mitochondrial inner membrane. So if we were to draw, right, a mitochondria, a really simple, right, this would be my outer membrane, right? Then I have an inner membrane. This is the matrix. And this is the inner membrane space, right? Y'all know mitochondrial structure, right? Yes, yes, okay. So what's going to actually happen is that we establish a gradient. And so what we'll do is and I'm just going to make it one box, right? The electron transport chain, it's really not one box, but you just have to roll with me, right? It's going to take hydrogen ions and pump them from the matrix into the inner mitochondrial membrane space. So it goes in this direction. And we're going to build up lots and lots of hydrogen ions in this inner membrane space.
Is it inner membrane or
Inner membrane
I'm
space?
sorry.
Inner.
Carry on.
Cool. So where do we have the highest concentration of hydrogen ions? In the inner membrane space. So how do molecules move? From high to low. That's diffusion, right? So what we're going to do is we are going to utilize that difference in proton gradient, and we're going to force those hydrogen ions to go through another complex. We're going to force those hydrogen ions to go through, this is the ATP synthase complex, right? And so when those hydrogen ions flow down their proton gradient, we are going to use that in order to drive ATP synthesis. So we're going to use ADP and inorganic phosphate, and we're going to make ATP. Am I drawing under the, no?
Oh,
No. Okay. So this is like the basic concept of how it's going to work. We are going to get into extensive detail on it, but it's really important to have a big picture before you get into the weeds, right? Okay, all ready? Everybody drew that? It's good to make yourself a little drawing. All right, so we're going to make a proton gradient, right? So in order to make that proton gradient, we have to have some complexes inside of that mitochondria that are going to actually be pumps of the protons. So what is going to pump the hydrogen ions? What system? What has to be the pump? ATP synthase is not a pump. It's not a pump because hydrogen ions are going from high concentration to low concentration. Where are hydrogen ions going against their concentration gradient? The electron transport chain. That's right. So the proton gradient is initiated by outward pumping by the electron transport chain. How is that electron transport chain going to do that? It's going to harvest the energy that are in those NADH and FADH2 molecules, right? We already said they're high energy molecules. So we're going to use oxidative phosphorylation in order to drive ATP synthesis. So in the electron transport chain, we have three major protein complexes. Complex 1, complex 2, complex 3. But you're going to learn the enzymes that are in those complexes. And that's what I'm going to want you to keep saying, because you can say 1, 2, and 3, but it's more important to know the names and what they're doing, right? And then when those inward flow, right, this inward flow is from high concentration to low concentration, right? By the electron transport chain is the opposite. This is from low to high. That's why it's a pump. Whereas this one is using what? Uses the gradient, right? Diffusion. Okay. All right, so what we have been calling the electron transport, now your book calls it the system. I have been calling it the electron transport chain for as long as I can remember. I'm going to call it the chain. All right, so be aware that the book calls it the system. All right, so the electron transport chain is really a sequential bunch of redox reactions that occur in these complexes that are embedded in the intermitochondrial membrane. What did we produce from citrate? What energy carrying molecules did we produce? NADH, what else? FADH2 is going to be oxidized to form what? FAD, right? Now remember when I told you that your terminal electron acceptor, what is that terminal electron acceptor?
Oxygen.
That is oxygen. Why is oxygen the terminal electron acceptor? When you looked it up in that table, come on, right? The conversion from O2 to H2O, what did you get? It was positive, right? 0.8 something. NADH and FADH2, what do you think those are going to be compared to our potential? They're going to be negative. They're going to be very negative. But we're not going to make one step, right? We don't go from FADH2 straight to O2. We're going to make 10,000 teeny tiny movements of those electrons. Those electrons are going to get shuffled around so many times. But here's the great thing. Every time we shuffle some electrons, and we usually shuffle them in pairs, there are some stages where we shuffle them individually, but for the most part, it'll be in pairs, that we're going to be able to move hydrogen ions, right? So we're going to use that sequential movement of electrons and that transfer of energy to pump the hydrogen ions into that intermitochondrial space. And the very last acceptor in that whole chain is O2. Okay. So that's why it's so important to understand those reduction potentials in the last chapter, because that's what all of this is based on. All right. So this is a slightly better drawing of the mitochondria, right? So in this case, this is just the inner mitochondrial membrane. The outer mitochondrial membrane is here. This is the outer membrane, right? So what's actually happening is it's really a circuit. It's really a circle. So you have to kind of think about it that way, right? So we have hydrogen ions in the matrix, right? And we call this the, whatever you're designating it, you put an N, right? And then you put a P for the inner mitochondrial space. And because this is more positive, that's the P. This is more negative, that's the N. So it's really designating the charge. So let's talk about why they call that the negative and the positive side. So if you look in the matrix, this is where we are, right? This is matrix. What did I make in the matrix that's energy carrying? FADH and A. Okay, these are my two energy carrying molecules. And so what they've done here is they've just represented them as an electron, right? And so we're saying that we're taking those electrons from FADH2 and NADH, and we're going to pass them along. And remember, this is not just one step. This is actually going to be tons of little steps, right? Until we eventually get to oxygen. And each time we transfer some of that energy, we are going to pump hydrogen ions from the intermitochondrial space to from the matrix to the intermitochondrial space. So that you don't have to write matrix and intermitochondrial space all the time. We say that these hydrogen ions are on the inside and these are on the P side. Just makes it easier, right? So now, once we're pumping them against their concentration gradient, they actually build up over here. So you can put yourself, I like to visually see that. I like to see, oh, I've got all these hydrogen ions on this side, right? And that's why they've made it red. So we have a very high concentration of hydrogen ions here. Well, what do hydrogen ions control? pH. So not only do we have a concentration gradient, we also have a pH gradient. And because a proton is charged, we also have an electrochemical gradient. So where we have lots of protons, we have a higher positive charge. Where we have less, we have a lower positive charge. It's not really negative. It's not really negative. But just like membrane potential, it's not really negative. It's just less positive. Correct. It's relatively negative, but it's not negative. Yeah. Could it be more confusing, right? So what happens is once these pile up, and once you have a high enough amount of them, they will run through this ATP synthase complex. And in the process, when they move through, that will drive this ATP synthase complex to convert ADP and PI into ATP. So we have this complete circuit, right? Because this hydrogen ion can then now be pumped back into the intermitochondrial space, right? So it continues as a circuit. So your book equates this to an electrical circuit, right? So you have something that is the resistor, the ATP synthase. If you don't know electrical systems, right, a resistor is whatever does the work, right? So the work is to make ATP. The capacitor. The capacitor in the system is something that temporarily contains the energy, right? And then you have the battery. Right, the battery is the energy. But your proton gradient is temporarily housing the energy that was in those electron carrying molecules, right? That gradient provides energy by driving ATP synthase complex. I just love this stuff. I think this is so interesting. I hope that y'all get that. So this is really cool. But sometimes you don't want to make ATP. Why would you not want to make ATP?
If you have enough of it already.
If you have enough of it already, and if you want to generate heat, so you can actually uncouple these reactions and give those hydrogen ions a different way. So let's do it this way to get back into the matrix. This is uncoupled, PLE, uncoupled, right? So ATP synthesis is not going to happen because these hydrogen ions are going through a different channel that does not create ATP. And so it uncouples it. And this leakage, that's what they call it, a leakage of protons, generates heat. So if you look, oh, and watch the animation. The animation's awesome. Watch the animation. If you look, this is an example, right? Here's our proton circuit. We have those hydrogen ions. We pump them into the intermitochondrial space. We make a lot of them. They flow down through the ATP synthase complex. We make ATP, right? This is the normal circuit. Now those hydrogen ions can go back to be pumped again and again and again. So here's an uncoupling. So this particular protein can be expressed in a particular cell type and can result in, instead of generation of ATP for work, generation of heat. So this is an example of brown adipose tissue. Right? So, for example, hibernation animals, right, will have brown adipose tissue that will generate heat while they hibernate. It's really cool. Now, that's a natural way that we can uncouple it. We also have antibiotics that can uncouple, but in this case, you don't have an additional protein being expressed, right? Our antibiotics are small chemicals, right? So actually what they do is they bind to the ATP synthase complex and they stop the ATP synthase complex from working. So now you're going to just build up this gradient. Do you have anything that can go through and be pumped again? No. Is your ATP synthase complex going to work? No. You will stop making ATP. What happens? Cells die, right? And you can target this to particular bacteria and things like that. But two ways that you can have uncoupling. So this is an example of an inhibitor, right? Whereas this is an endogenously expressed channel protein. Right. Okay. So I want to tell you something interesting about your turkey. Right? So this, we just had Thanksgiving, right? And I know everybody has a preference for dark meat versus light meat, right? It doesn't matter what it is. Everybody has a preference. You know you're going for that leg, or you know you're going for the breast meat. You have this particular one that you like, right? So what is the difference? Why is some meat dark and why is some meat light? It depends on the concentration of mitochondria in the cell. The more mitochondria you have, the darker the meat is. Isn't that cool? So depending on what the muscle's function is, it can be a fast twitch or a slow twitch muscle. Did y'all learn about this in physiology? No? Okay, so
You learn about it in comparative work anatomy.
oh, you do? That's awesome. Okay, so if you go hunting and you shoot like a duck, what is all the meat color? It's all dark, right? Even the breast meat is dark. Why? Why is a wild animal have more dark meat than a farm-raised animal? Farm-raised turkeys, do they fly? So how much do they use that breast meat? They don't. So they don't have a whole lot of mitochondria in them. So the difference in color of dark meat and light meat for wild game versus farm-raised food is different. Things you never knew you wanted to know, right? So for Christmas, what you can do is you sit down at the table and you say, I'd like a piece with more mitochondria, please.
I love it. Look at me like I'm crazy.
Right, how to find the nerd in the family, right here. It's right here. Okay, so back to the real world, right? So here's an example of a mitochondria, right? We have an outer membrane, and then we have that inner membrane. But what's different? What do you see that's different between the outer membrane and the inner membrane? That inner membrane is very convoluted, right? Okay, so what do we call that? Convolution. The cristae. So the cristae. So when we talk about the electron transport chain and the ATP synthase complexes, each one is like a teeny tiny dot on here. So why is the inner membrane convoluted where the outer membrane is not? The outer membrane is not housing any part of the electron transport chain. It is only in the inner membrane. So we have a ton more what because of the convolution? Surface area, space, right? Okay, so I did the math on this. I just can't help showing the nerd in me today. So I did the math on this. So you usually have 10 to 100. You don't have to write this down. This is just interesting. You have 10 to 100 mitochondria per cell, right? So it depends on the cell type, but you know, we'll ballpark it. So if you have surface area, that gives you about 5,000 micrometers squared for surface area. CRFA, surface area per cell. That's pretty good. That's actually pretty good. But in a form that's more understandable, right? How many cells do we have in our bodies? Yeah, a lot. What's a lot? 10 to the 13 to 10 to the 14. So it's in the trillions. Trillions, right? The trillions. That's a lot. Okay, so that gives you 185,000 meters squared. That's pretty good, right? So what is that in terms of surface area? That is 26 full-sized soccer fields in one person from mitochondrial matrix surface. What's that?
Really thin layers, though.
Really thin layers. They're really, really, really thin layers. Really thin. But that just tells you how many electron transport chain systems you have in your body. How much of your cell is devoted to making ATP, right? That's a ton. Isn't that cool? I might think that this stuff is a little too cool, but that's okay, right? All right, so we said that the electron transport system contains 3 major complexes, right? Okay, well, when they were studying the electron transport system, what they did was originally the way they named these is by what was discovered first, right? So we do have 3 major complexes, but when they were named, they were given complex names that don't correlate exactly to function, right?
It's the photosystems all over again.
It's like the photosystems all over again. It is. It is. So technically there are 3 major, but you have to kind of know them all. So complex 1 is the NADH ubiquinone oxioreductase. So when you're learning these names, what's important to understand is you can look at the name and you can know what's happening, right? So can you guess we're taking electrons from where? NADH, and we're going to transfer them to ubiquinone. I mean, you know, so yes, these names are kind of get crazy, but we're going to talk about the function of each, and then it's going to make sense. So they did name these well, and we'll talk about each of the individual functions. So complex 2 is succinate dehydrogenase. I think we've heard that before, right? Complex 3 is ubiquinone cytochrome C oxioreductase. Complex 4 is not technically a complex, but it's cytochrome C oxidase. So, and then the very last one that they discovered was the ATP synthase complex. So we don't really call it complex 5 anymore. We just call it ATP synthase complex, right? So when we look at these initial complexes here, these are embedded in the inner mitochondrial membrane. But it's important that they have functional domains that face towards the mitochondrial matrix, because where is NADH? In the matrix. Where is my final electron acceptor? In the matrix. So you have to have functional components for all of these that face the matrix of the mitochondria. Yeah? Okay. So here's the real electron transport chain, you know, not one box, right? So when we go through citrate cycle, right, we take NADH. That NADH is going to go into complex 1. So you remember when I showed you that breakdown diagram and I said you can generate more ATP from NADH than you can from FADH2? Do you remember that? So why is that? Well, NADH's electrons enter the transport chain early. They enter at complex 1. Look at FADH2. Where does FADH2 drop off its electrons? To complex 2. So if we have FADH2, we've missed complex 1. And complex 1 is responsible for pumping 4 hydrogen ions from the mitochondrial matrix into the intermembrane space. So we've lost that, right? So we're going to end up pumping fewer hydrogen ions and therefore making less ATP. Okay, so if we start from the beginning, we have our NADH, we've dropped those electrons off at complex 1. Complex 1, as this blue line is really, it's a complex. So it's really electron hopping, right, along, and as those electrons are passed along, that's what's pumping that's what's giving the energy to pump the hydrogen ions from the matrix into the intermitochondrial space. So we move from complex 1, it gets transported to complex 2. Complex 2 does not pump hydrogen ions. We have to move our electrons from complex 2 to complex 3. Now, when we get to complex 3, we're going to transport 2 hydrogen ions here, and we picked up 2 hydrogen ions here. So the combination of these 2 and these 2 will be pumped into the intermembrane space. So we pumped 4 here, we pumped 4 here if we're starting with NADH, right? If we're starting with FADH2, really, we've only done 2 so far, okay? All right, so now we go from complex 3, we're transported 1 electron at a time. This is, remember I said most of the time it moves by pairs of electrons. Here we actually have to split up the electrons and we have to do it twice. So cytochrome C can only carry 1 electron at a time over here to complex 4. So complex 4 is where we're going to eventually end up transferring those electrons to O2 to create H2O, right? And so here we're going to transport another 2 hydrogen ions. So how many total hydrogen ions did we pump into the inner membrane space? 4 here, 4 here, 2 here, right? So now, what happens to all those hydrogen ions? They're going to build up and they're going to flow down their concentration gradient. So for every 3 hydrogen ions that get transported through the ATP synthase complex, we're going to take 1 ADP and 1 PI, and we're going to turn it into 1 ATP. But where's the ATP? We just made it, but where is it? It's in the matrix. Is that where we want our ATP to be? No, we got to use it in all parts of the cell. So we have to ship this out. How do we ship it out? We're going to have to ship it out with transport proteins, right? So at the same time that we're transporting out a molecule of ATP, we're going to transport a molecule in of ADP. So what kind of a transporter is this? It's an antiporter. This one's an antiporter. Okay, now we know we got ADP. That was half of what we need, but what else do we need? We need inorganic phosphate. So the inorganic phosphate will actually travel, 1 inorganic phosphate because what's the charge on an inorganic phosphate? It's negative. So we can transport it with a positively charged hydrogen ion that's flowing down its concentration gradient in what kind of a transporter? A symporter. And we can use that energy to push that negatively charged inorganic phosphate. I mean, that's you know, that's not it's not going to happen naturally. So then we can generate all the components that we need for ATP. So some of the hydrogen ions are used in ATP synthase complex, and some of the hydrogen ions are used in translocating that inorganic phosphate. Yeah, it's cool stuff. All right. So go back to the beginning, right? Y'all ready? Yeah. Okay. So let's go back to the beginning. We have NADH oxidation, right? So we're at the very beginning of the electron transport chain. We're at complex 1. This is going to take place on the matrix side of the inner mitochondrial membrane. So you made NADH in the citrate cycle. It's therefore in the matrix. So that's where you have to start. So you are taking 2 electrons from that NADH, and that's what's going to initiate redox reactions. And then at the end, eventually, when we get to complex 4, we're going to have oxygen being reduced to water. But that's not until complex 4. So the other way that you can start is to start with FADH2. If we start with FADH2, where are those electrons going to enter? If we start with FADH2, where are those electrons going to enter? Complex 2. So at complex 2. Here, 2 electrons, NADH occurs in complex 1. Okay, we have that, right? So really inside of these complexes, you have multiple electron carriers inside of each complex. And those electrons move from electron carriers that have negative potentials to ones that have more positive potentials, right? Because if you look at that table, FADH2 and NADH are basically at the bottom of the table. And so all the other stuff in between in that table in your book are all the parts of the electron transport chain. And that's how we're going to drive that proton pump. Okay. So before we talk about the details on that, because I really don't want to go too complex, because I feel like you're going to forget it when you go study for your exam, right? I know you are. I know you are. Okay, so let's do this instead. Let's talk generally about oxidative phosphorylation, because if we get into the nitty-gritty weeds, we're just going to have to do it again, and I don't want to do that. So why is oxidative phosphorylation, what does it do for the cell? Why do we need oxidative phosphorylation? We need to generate ATP. Because we made a little bit of ATP, but we didn't make a lot of ATP, right? So we're going to generate ATP. What are we generating it from? From products of, that's okay, citrate glycolysis, right? So if we look at, let's try to build the overall net reaction of oxidative phosphorylation, right? Because we know where our substrates are coming from. So don't worry about the coefficients yet. Let's just talk about the pieces. So what are the products from citrate and glycolysis that we're using to undergo oxidative phosphorylation? NADH. What else? Okay, so an FADH2. So let's pretend that we're just starting at the very beginning of the complex, that we just had NADH. Okay, let's go with that assumption. So what else do we need when we transfer these electrons that is going to pump what molecule? What are we, the electron transport chain is a pump. That's what it is. What does it pump? protons, hydrogen ions. So we're going to have hydrogen ions, right? And oxidative phosphorylation. The phosphorylation part of this is ATP synthase. So what do we need in order to make ATP synthase work? I know I want to make, I'm going to put that as my yield sign, and I'm going to put the products down below. I know I want to make ATP, right? So what do I need in order to build ATP? ADP, inorganic phosphate, and I'm missing one other thing. What am I missing? What else? Yes, at the end of the electron transport chain, I have to have oxygen. So then let's look at each individual thing and see what it turns into. So what does the NADH plus the H plus turn into? NAD plus, right? We regenerated it. Where are we going to use NAD plus? And well, yes, you can use it in glycolysis, but typically that NAD plus is regenerated in a different way for glycolysis. So this NAD we're going to really use very quickly for citrate, right? So you can see how the pathways are all connected. Okay, and what about water? We don't end up, oh, I just gave it away. Oh, man. What about the oxygen? What does the oxygen turn into? It turns into water. Oh, man. I hate when I do that.
I actually ruined the joke with that by accident. I accidentally did it, so I was like,
Yeah, I do that. I do that pretty regular. All right. So start to learn the names of the different systems. Like, I know you're going to go home and study for the exam on Friday, and you should. So then over the weekend, go in and start to get familiar with the names of the different complexes so that when you get to class on Monday, I'm not speaking a different language. You know?
We're gonna be doing that anyway.
Well, yeah. Yeah, so Sunday night. All right, y'all have a good afternoon. And you'll have a different appreciation for that dark and light meat.
Yeah.
So funny. Anybody on Zoom need anything?
Okay, can you go back to what we were talking about at the very beginning of class?
Yep. So does anybody on Zoom need anything before I log off? Okay, I'm going to go ahead. I don't hear from anybody, so I'm going to go ahead and end Zoom.